Yes, I really. Wow.
I think you need some deal, bro. 很臭嘞。
How should I fix my smelly armpit?
Glycolic acid, the most hackable viral ingredient in all of skincare. This exfoliator not only breaks up skin cells, kills bacteria, but also stimulates collagen growth, allowing it to help with melasma, dark spots, pores, acne, and maybe even body odor. So here's how to use it. Once nightly, apply to clean skin.
Okay, let me try.
So have you ever wondered why armpits actually smell like Ming Kwang's armpits? Maybe you think it's because of the sweat that you produce, but contrary to popular belief, sweat itself is actually odorless. To understand our research, let's take a closer look at what's happening on a biological level. So the human body has two main types of sweat glands, eccrine and apocrine. Eccrine glands are spread across almost the entire body, while apocrine glands are concentrated in areas such as the axilla, which is our armpit. Apocrine secretions are rich in proteins, lipids, and steroids, and are initially odorless. So, however, odor arises when resident axillary microbes, primarily Corynebacterium and Staphylococcus, metabolize our sweat components into sulfur-containing compounds and short-chain fatty acids. And these metabolites are actually the ones that are responsible for that characteristic body odor. Additional factors such as pH changes, hygiene practices, diet, or the use of antimicrobial products can also disrupt this balance of the microbiome, causing dysbiosis and promoting the overgrowth of odor-causing species, altering the chemical profile of our sweat and intensifying body odor. Traditionally, people use deodorants, which are antimicrobial, or antiperspirants, which limit sweat secretion. Recently, however, glycolic acid, which is a compound well known in skincare as an exfoliant, has gained popularity as a DIY deodorant. But what exactly is glycolic acid, and why might it work in this context? Glycolic acid is a small alpha-hydroxy acid, also known as AHAs, and it is widely used in dermatology for its keratolytic and exfoliating properties. At acidic pHs between 3 and 4.5, glycolic acid exhibits antibacterial activity in vitro, particularly against gram-positive bacteria, and these include our Corynebacterium as well as Staphylococcus. This suggests a potential mechanism by which glycolic acid could actually reduce this odor-causing bacteria in our axilla. However, the problem is, despite all the hype, there is very little clinical evidence that glycolic acid really works in reducing body odor. And so this leads us to our research question. How does glycolic acid affect the abundance of odor-causing bacteria in the axilla microbiome? Thus, through our experiment, we would expect to see a reduction in Corynebacterium and Staphylococcus, and in turn, other microbes would then increase in their relative abundance. Studies demonstrate increases in Cutibacterium, Acinetobacter, as well as Pseudomonas, which are more acid-tolerant and are capable of utilizing alternative substrates. Thus, they are able to occupy ecological niches which are left vacant by the decline of dominant odor-producing species such as Corynebacterium and Staphylococcus. And this shift reflects rebalancing rather than true dysbiosis of the microbiome. However, chronic or excessive disturbance of the microbiome through excessive treatment of the area could also lower the diversity of the microbiome itself and favor opportunistic colonizers, potentially leading to mild irritation.
The intervention that we've chosen is the ordinary exfoliating toner, which contains 7% glycolic acid with an acidic pH value of 3.6. We applied 2 ml of the solution on a cotton pad and placed it onto one of the armpit for 30 seconds to allow the glycolic acid to make contact with the axillar skin. We will then air dry the axilla for 2 minutes before repeating the same procedure for the other armpit. A 2 ml volume was used as it was sufficient to wet the entire cotton pad, while 30 seconds of contact time was chosen as the manufacturer instructed to limit the contact duration. The intervention lasted for a total of 9 days, with the glycolic acid solution being applied every other day after the evening showers. As this is our first experience using glycolic acid on the axilla, we based the frequency of application according to the manufacturer's recommendation.
We collected our samples 2 days before intervention start and 1 day after the final intervention, both before evening showers. Axillary swabs were collected from both the left and right arm. This was done in a combination of vertical zigzags, horizontal zigzags, and circular motions. The sequence was then repeated for 3 times on each arm to collect a total of 10 samples for pre-intervention and another 10 for post-intervention. Our method ensured the swab covered a larger area of the axillary region and increases the likelihood of biological material collection. Collecting from both arms contributed to an increased representativeness, and we were also able to account for inter-individual variation in sweat deposition, which could potentially impact microbial growth, as the handiness of a person can influence axillary sweat secretion. However, as our swab collection was not done by a single individual, there are inconsistencies in how the procedure may have been followed, so that a lesser or greater area of the axillary region was covered. Furthermore, force applied may also affect the amount of DNA that can be collected. Overall, these can contribute to an increased variability in the sample collection method.
Next, let's move on to some of the additional controls for the experiment. So firstly, we limited deodorant usage, and we had a 4-day washout period which started on 19th of August before the start of the experiment. This is because deodorant may contain antimicrobial agents which can act as a confounder, and by preventing deodorant usage, we can prevent this potential confounder and result misinterpretation. We also had daily axillar shaves to increase the glycolic acid to skin contact. Next, we also showered twice a day in an attempt to mimic Singaporean behavior, as well as for general hygiene purposes. We recorded the timing of the interventions as well to ensure a consistent period of exposure before the glycolic acid application.
One limitation of our study will be the small sample size. With only 7 participants, the dataset may be too small to detect statistically significant differences. With a small dataset, random variations are magnified, making it difficult to determine whether the changes in the relative abundance of Corynebacterium and Staphylococcus species reflect the true effects of the intervention or are simply due to chance, potentially resulting in non-significant findings. Additionally, results from this study will not be representative of the broader population. Future studies with larger sample sizes and more diverse participants are needed to validate these preliminary findings. The second limitation of this study is the variation of sweat production between individuals. Sweat levels can be affected by hygiene habits, lifestyle, and environmental factors such as temperature and humidity, and these differences may alter moisture, nutrient availability, and local skin conditions, thereby influencing bacterial growth independently of the intervention. High sweat production can promote the growth of odor-causing microbes such as Corynebacterium, which thrive in environments with higher concentrations of sweat and sebum. Individuals with high sweat production may experience reduced efficacy of glycolic acid, making it difficult to attribute observed changes in bacterial abundance solely due to glycolic acid application. Some possible remedial measures to reduce variability from sweat production would be to standardize participants' routines, inform participants to avoid intense exercise or activities that increase sweat during the study period, standardize bathing and clothing practices to minimize differences in sweat accumulation, monitor sweat-related variables such as tracking participant activity and temperature exposure to account for differences in sweat production during analysis. To minimize inter-individual differences in sweat levels, aluminium-based antiperspirants can be applied to temporarily block the sweat pores at the skin surface. However, we need to account for the fact that antiperspirants themselves may affect the microbiome, since reduced sweat may alter skin moisture and nutrients. In future studies, we plan to design 4 groups: a baseline group to show natural microbiome changes without intervention, a glycolic acid-only group to see its effect under normal sweat conditions, an antiperspirant-only group to observe the effect of reduced sweat, an antiperspirant plus glycolic acid group to show the combined effect. Comparing these groups will allow us to isolate the effect of glycolic acid while controlling for sweat variability. Lastly, individual health conditions such as eczema can influence baseline skin microbiome composition, such as causing reduced bacterial diversity and dominance of S. aureus. Since the baseline skin microbiome may already be altered, the changes in response to intervention may be different from people without eczema. Future studies could mitigate this by screening participants for skin conditions, recording the samples that are from participants with flares in the axilla. Participants with skin conditions can be excluded from the study to reduce baseline variability. If included, stratification or subgroup analysis can be performed to analyze their results separately to check if the response differs from healthy participants and ensure that the underlying skin conditions are not a confounder in the study outcomes.